I hope everyone enjoys today's conversation about digital innovation for cell and gene therapies. My name's Trent Carrier. I'm the Chief Operating Officer for L7 Informatics. L7 is a company focused on digitalizing life sciences. We have a booth, so encourage anyone who's interested in learning more to stop by and learn what we're doing. Today is my pleasure to introduce our panelists. Julien Cottineau is the co-founder and chief science officer at WhiteLab Genomics. Tim Liu is the co-founder and CEO of Senti Bio. We have Kevin Gordon, chief digital officer for Biotech, and Fabian Gerlinghaus, the co-founder and CEO of Cellares Corporation. I practiced that last night. So we'll just take a couple minutes, and in that order, I'll let our speaker, our panelists introduce themselves and talk a little bit about their companies.
Okay, cool. Should I start?
Yes, please.
Yeah, okay, perfect. Good morning, and again, thank you for the invitation. Very pleased to be here. So I'm Julien, I'm co-founder of WhiteLab Genomics. We are a startup based in Paris, specialized in AI for gene and cell therapy. So basically, we provide different applications for discovery and design for gene and cell therapy. So we do have different technologies that we used to actually de-risk and accelerate the development of the different products. We work either with academics, biotechs, and pharma all along. And so we are created like 4 and 5 years ago, approximately. We're a team about 25 people. We were lucky enough last year to actually finalize our fundraising. We raised 10 million euros in Europe, and we are really happy to have our invest supporting us so far, and absolutely pleased to actually develop different applications for either AAVs and also lentivirus and non-viral development. The idea behind that is really to push forward the boundaries of what the strategies that have been developed so far. Our approach is really based on a guided and rational design. Basically, what we do is we understand the human body, identifying different receptors that allow us to actually move forward with a more specific and more rational design. So the idea behind that is to develop specific peptides that will fit with receptors that have an expression pattern specific to the cell type, so we're going down in granularity of the different tissue in order to actually back to the design to push forward the boundaries of the specificity of vector delivery. And so the other application we developed is really like the payload design itself. What we understood and what we want to push forward is really the association of the two components, either the vector and either the payload, to actually increase the specificity and the efficacy of gene and cell therapy.
Great. Thank you very much. Tim.
All right, good morning, everyone. I'm Tim Liu, co-founder and CEO at Senti Bio. Senti Bio is a company based out in South San Francisco. We are focused on developing drugs and bringing them to clinical trials and into the market eventually. Really at the core of what we do at Senti is trying to program living cell therapies with greater quantitative and precise activity. So as an example, our oncology programs are really focused on this idea of introducing logic gates into living cells so that instead of a cell therapy that only targets a single antigen, of which there's some out there that are great, but there's many cancers that can't be accessed just with a single antigen. We can build products that have 2 or 3 CAR receptors, for example, on the surface, and we've designed these CAR receptors to interact with each other in such a way that we can achieve, for example, targeting of cancer cells while explicitly protecting healthy cells. So this ability to activate and suppress is some of the quantitative control we've been able to build into our cell therapy products. In addition to that, we do design what we call gene circuits for a variety of other applications. We have an ongoing collaboration with Spark, building engineered promoters, essentially, for AAV gene therapies, where we are essentially designing synthetic compact promoters to achieve 10,000-fold or more specificity for your desired on-target cells versus your off-target cells to really drive a wider therapeutic window. We also have a collaboration with Blue Rock, programming iPSCs to control their phenotypes, both in the in vitro or lab-based phase of those products, or in vivo, and ultimately for eventual human use. So we're really excited to be able to start pushing these sort of products towards the clinic. We think the opportunity to, instead of relying on single static type therapies, we can really take advantage of living biology and program more dynamic, responsive sort of products in vivo. So appreciate the time.
Super. Thank you, Tim.
And Kevin Gordon, I clearly failed at life and I'm not a co-founder of anything. But appreciate everybody being here this morning. So I'm the Chief Digital Officer of Ori Biotech, and Ori is a platform company working to industrialize the manufacturing of these amazing curative therapies. As a plug, we'll be doing our company presentation later today, so stop by if you want all the details there. My claim to fame is actually coming previously from a place where we built the fully digital record for CAR-T as well as Lenti manufacturing through the FDA for paperless with release by exception, and it's been fun because recently some of the FDA's new multi-therapy in one ballroom is a lot of what our Type C filing was. So really interested in the digital innovation in this space and working on driving those things forward.
Thank you, Kevin. Fabian?
Good morning, everyone. Fabian Gerlinghaus, co-founder and CEO of Cellares. At Cellares, we're on a mission to meet the total patient demand for cell therapies globally. We're the world's first integrated development manufacturing organization, or IDMO. IDMOs overcome the limitations of conventional CDMOs really through technology integration and vertical integration. I'll be going into more detail on all of this in my presentation on Thursday, but in brevity, on the technology integration side, we've designed, built, invented, tested a fully automated high-throughput cell therapy manufacturing platform called the Cell Shuttle that can actually produce up to 16 cell therapy batches simultaneously. And importantly, it brings down the amount of facility space and labor required by about 90%. And we're now proceeding to deploy and operate cell shuttles in IDMO smart factories around the world. We've got one in South San Francisco, another one in Bridgewater, New Jersey. We're breaking ground on the European facility next year. And because of the 90% reduction in facility space and labor required, our IDMO smart factories actually have 10 times the productivity of a conventional CDMO facility, meaning with the same amount of people and the same amount of floor space, we can produce 10 times as many cell therapy batches while reducing the cost per patient dose by about 50% compared with conventional CDMO pricing. And very exciting update. We've got a few partners that we're working with. We recently announced that Bristol Myers Squibb partnered with us. They're giving us a key CAR-T cell therapy asset to automate and transfer onto the Cell Shuttle. Bristol Myers Squibb also participated in our massive $255 million Series C that we recently announced. And just this morning, Endpoints broke the news that Bristol Myers Squibb is giving us a second CAR-T cell therapy. So we signed a second deal with BMS to automate and transfer a second CAR-T cell therapy onto the Cell Shuttle. And if you're the leader of a cell therapy company and you'd like to do the same thing, please get in touch with my biz dev team. We've got quite a presence here this week, and they'd be happy to meet with you.
Thank you, Fabian. So our panelists, as you can see, represent DNA to factory scale considerations, and those are really, as we all know, the key challenge for cell and gene therapy. I also just take a pause and congratulate all of you on your fundraising successes. It's not an easy market, and I think, I'm sure digital innovation was part of your company strategy when you pitched it. So for all of you who are also looking to fundraise, it sounds like you could take some lessons and hints from these guys. So I'll start with some Q&A. I'll start, Julien, if you don't mind. You witnessed the evolution in genomics research. How can adopting a fully digital platform de-risk and accelerate gene therapy development through genomic data analysis?
Thank you, Trent. Absolutely. We actually, the idea at the very beginning is really we observe, the trial and error strategy that have been done in the lab for, 40 years, something like that. And all the idea behind is we do generate all along our story, our experience in labs, a massive amounts of data. And the idea behind all of that is, do we, able to build models to all those data in order to actually find some hypotheses that we can work on, reduce the time of development, reduce the money that we spend all along to test all those hypotheses. And the idea behind the development of WhiteLab was really this: how to change this trial and error strategy to actually find and pause a bit a moment and saying, okay, what should we focus on? And basically, how can we going out from one gene, one patient, one gene therapy, one treatment? And so the idea behind all of that was really basically, can we integrate the different nature of data, genomics, the structural protein information, but as well as, bioprocess, development, and integrate this network of information in order to guide all along the development of the gene and cell therapy. And so what we observe in all of that is actually people are really keen to move forward with this strategy. And the idea, they don't know that they do have such amounts of generous information in their hard drives. They don't know what to do with that. And actually, even, the failure data are super informative, because we can drive and the models that to understand, either what are, the good path to actually work on, but also what parameters we should definitely not consider because they do not influence the overall process. And, all this information linked together, we do have absolutely good results all along to move forward with this idea. I don't know if our colleagues actually shared those ideas, but I think that all the field is now ready, to change, to evolve, to accept, the AI in general. And the thing is, it's really important for me, I guess, that we will discuss that in the future, but the nature of that, how to network it in order to, to push the boundaries about what have been done so far.
Great, thank you.
Maybe I will stop here.
Yeah, thank you. Kevin, Oreo Bio is known for a cutting-edge approach to cell and gene therapy manufacturing. Can you elaborate on how a digital platform helps you improve control and regulatory compliance for cell and gene therapies?
Absolutely. I think it's thinking about quality control and digital and sort of the journey that patients go through in this space with getting enrolled in a hub service, and really, as they go through the early stages of getting apheresed, they get a really detailed, almost Uber-like view of what's going on with their cells. The moment it lands in a manufacturing plant, that goes dark for a period of time in 99% of the situations today. And so at Ori, having a digitally native connected platform manufacturing those cells, one of the things that we're really interested in doing is providing visibility and sort of transparency back out to the larger audience on those pieces. Has a few benefits. One, a digital record that runs from apheresis back into patient gives you complete traceability of really what's going on. And then two, you actually could potentially, and as we've seen sort of case studies of, reduced vein-to-vein times for patients in the space. So taking what was a set of unit operations, digitizing them, potentially giving them the opportunity to pass into the larger patient record really is a big step forward in control and actually can help with both quality and compliance.
Great, thanks, Kevin. Tim, your company's focused on synthetic biology. How do you apply digital innovation in the design and engineering of new genetic circuits for cells?
So the reality of it is biology is actually not digital. It's not evolved in a way that's very modular or easy to understand. So I think in the early days of this field of synthetic biology, people tried to go solely with the approach of, let's force this idea of digital computing onto biology. And I think what people realize is through that process, you have to be very explicit about trying to essentially make modules in biology if you want to be able to click things together. That process of sort of building modules, clicking them together, and hoping that they work well together can get you to a certain extent, some function, but it's not necessarily the way to get to the optimal function. And so I think at Senti, we've realized that sort of the best approach to this, at least for the time being when we can't simulate everything that happens in the cell, is essentially a hybrid approach. One is we do spend a bunch of time up front trying to modularize or get rid of as much of the unwanted sort of biological interactions inside the cell as you can, right? That's never completely possible, but if you start off with a basic approach there, that helps you simplify the problem. And two is, what Julien mentioned earlier, is really then incorporating a lot of these high throughput strategies to then further optimize and take you to that next order of magnitude of performance that you're looking for. So what we've been doing over the last few years, in large part, is really developing high throughput, massively parallel assays for what you want these cells to do, whether it's activate on cancer cells, protect healthy cells, or proliferate upon presence of a signal. These sort of quantitative assays take a lot of time to define and build well so that they are accurate. You have to make sure you have the right controls in there to do so. But essentially, if you can convert these high throughput assays to some sort of sequencing-based assay, you can boost the throughput of these assays by orders and orders of magnitude. And once you do that, you can collect much more detailed quantifiable information about how these million different gene circuits perform, each on their own. And then from that, you can then start applying some of these more recent digital innovations around computational models, machine learning models, to then suss out what are some of the designs in here that worked well and did not, and then from those patterns design sort of next rounds. So as an example, when we've gone through and designed promoters for AV, that first round of design was partially hypothesis-driven, partially driven by whatever patterns we can suss out from the public data sets. And then we've gone through now 3 rounds of iteration to get to promoters that are, like I mentioned earlier, 10,000-fold or more specific for a given target, as stronger than any of the known constituent promoters out there today, and also very compact. And so that's where this iterative design can be really important. But you have to start out with at least a reasonable hypothesis. Otherwise, you might be, I think, just exploring in the dark. And you need to be able to iterate through large data sets pretty quickly.
Great, thank you, Tim. Fabian, at Cellares, you're at the forefront of bioprocess innovation. How are you approaching digital innovation to manufacturing processes and equipment?
The main point that I want to make is that digital innovation doesn't exist in a vacuum. Digital innovation needs to be integrated with hardware innovation. And the core software stack that we've built at Cellares really covers 4 big areas: process design, digital process design, digital process execution, digital process monitoring in real time, and then reporting. That's electronic batch records, for example. And all of these things, you can't do that with a disjointed set of benchtop equipment where it's very hard to get data in and out because either they don't have software interfaces at all, or every vendor gives you a different API that doesn't actually allow you to get the data out that you really want if they measure it in the first place. So hardware integration is a key component here. And by means of example, the Cell Shuttle platform has the flexibility to support 90% of cell therapy modalities, both autologous and allogeneic, CAR-Ts, TCRs, and Ks, basically anything based on suspension cells. And that level of flexibility is achieved even though everyone uses the exact same hardware, same cell shuttle, same cartridge. We've built so much modularity into the hardware platform that we can then expose in a digital process design studio. We've got a video about that on the technology page of our website, where you see how process scientists can now do digital process development and literally click together workflows with the individual unit operations, configure their required reagents, all of the underlying process parameters. And that's what I mean. We've moved the entire process customization problem from hardware into software, yet the software and the hardware are integrated together. And that then continues as we're doing process monitoring in real time, for example. There's a lot of QC capabilities on the Cell Shuttle. All of that data comes out in real time, feeds into an integrated data stack. And this integrated data stack then can be used to do a number of very useful things. One of the primary things we're doing with this is we're moving the industry from paper-based batch records to electronic batch records. And more than that, we can actually auto-generate electronic batch records. Why? Because The entire cell manufacturing process is automated on the Cell Shuttle, the platform. The software knows what it did to the product at what time. It gets the readouts from the QC instrument. All of this ends up in an integrated data stack, which then allows our software platform to auto-populate electronic batch records. And this is how we're getting rid of 500 pages of paper-based batch records that today are being generated for every dose and constitute a massive overhead. So that's the core stack of the proprietary software, digital software platform at Cellares. That's neatly and intricately integrated with the automated cell therapy manufacturing platform. Again, process design, execution, monitoring, and reporting. And then beyond that, obviously there's a need to integrate with third-party software platforms. So we're integrating with the LIMS systems of the world, L7 for example. In ERPs, internally we're using SAP and we're integrating with SAP, the QMS systems of the world. And then from there, we're certainly going to expand and offer chain of custody, chain of identity solutions as well.
Thank you. Tim, of the panelists, you have one of the more dynamic data profiles given the kind of massive screens that you do as well as the depth of the data. Can you talk about how those different shapes of data are managed within your workflows, and then where do you fit digital technologies into those workflows?
Yeah, so when we look at building these gene circuits, number one, depending on the application, the assay is like completely different, right? So the assay for gene therapy, where we're trying to look for selective expression in your on-target cells versus your off-target cells, versus like a CAR-T or CAR-NK assay where you're looking at killing and you're looking at other maybe phenotypes like a flow cytometry and others. It's complicated, right? And so number one, there's a lot of custom assay development that's necessary for a given application. So we do have biology teams that really specialize in trying to do that, and like I said, setting up the right controls and quality control process in that whole step to really make sure the data we're pulling out is actually accurate, so that even though you converted it to a high throughput way, it's still representative of how you would have done it in a more sort of one-by-one careful fashion. Two is then sort of capturing and analyzing that data is not simple depending on what format it comes in, right? Some of this is imaging based, where we're looking at, let's say, our CAR-T or CAR-NK cells killing in a batch of, a sea of living and healthy cells. We sort of literally just take images over time and look at, are the healthy cells proliferating, are the cancer cells being killed, and what's the dynamics there, right? So there's a lot of image processing that needs to be done there to automate that so that some person doesn't look under the microscope, have to count everything. In other situations when we've converted our data format as NGS, then there's a lot of obviously processing that needs to come off of those sort of reads. I think the one area that would be really interesting that we're not as actively sort of being able to capture at this point is the linkage between the gene circuits that we're developing and the cells that they're in, and the ultimate process development step. I think there's still in our field a little bit of a gap between those two things where we are spending a lot of time looking at the functional assays, like what is the cell doing, is it achieving the desired therapeutic function? But a lot of things that we're modifying in these cell and gene therapy products ultimately also affect how they're going to grow or how they're going to expand, et cetera. And so I think building stronger linkages there could be really helpful in our process. So anyways, we've had to develop, I guess, custom readouts for many of our assays. And then the objective function for some of these design algorithms is really oftentimes multifactorial, right? We're not just optimizing for one thing. You might be optimizing for 3 or 4 things in parallel, and that's another part where it's tricky when you're trying to develop some machine learning algorithm to get you there. You're actually optimizing essentially towards a multi-objective function.
Great, thanks. And we'll come back to kind of how you manage those different scales, right? The DNA scale and the factory scale. That's the gap. So let's make sure we come back and talk about that. Kevin, a question for you is, what does the end state look like for cell and gene therapy manufacturing, and where does digital infrastructure play a role in that? And perhaps going back to Tim's comment, like how do you start building, what networks are needed to, in fact, have a full understanding of what you're doing?
Yeah, and I think the title of the panel, right, Redefining Manufacturing Through the Digital Protocol, is really sort of that exact thing, is redefining a lot of what good looks like today and going forward. And so I think looking into the future, one of the pieces that people have talked a lot about is distributed manufacturing or point of care manufacturing. And one of the pieces that we've been running quite a bit at Ori is to take a singular protocol, run it from a central command and control location across both UK, US, both in Ori facilities and with partners, and really start to prove that a singular protocol can be run like for like in different facilities by running a real digital batch on those things. But going forward, I think it's going to be, a continued challenge on what those things look like, and really what it's going to come down to is what's the driver to do it. And I think what we've been seeing is a reduction of cost of goods above 40%, a time of transfer of process into the platform, like 50% of what would be standard when you're going from paper to digital, and then really ongoing run costs that start to really continue to decrease because you're monitoring and managing these things as more of a centralized set of processes. And that digital protocol is really what's required, right? When Fabian talks about getting rid of those 500 pages of paper, those 500 pages of paper are expensive and painful to continue to manage. And we're in complete agreement that a digital one and powering by companies like L7 is really critical to where we need to be going to really start to be able to break down these giant singular manufacturing platforms and allow them to start to spread and get closer, faster, and cheaper for patients.
Great, thank you. And Julien, I'm going to toss to you an extension onto Tim's point, which is, in order to bridge these, scientific to IT challenges, what do you think needs to happen in terms of the bridges that need to be built, specifically to enable the types of visibility and data understanding across these different scales?
I think the pain point here is to be versatile. Versatile in order to actually understand the hypothesis, elaborated by the scientist, and also to understand the IT guy as well, because we don't talk the same language. That's the first observation we can do. And the thing all along the work we've done in WhiteLab, what we observe is we definitely need to have different profiles that actually can talk to each other and actually can understand, the different meaning or what a model means. It's totally different from a biologist to an IT guy or to a math guy. It's totally different. And so, for example, when we partner with different biotech, Cellares Biology is one of the recent partnership that we developed. For example, they do have this idea about, we do have a preclinical pipeline, we do have data, we do have data, and they do not know how to actually elaborate the hypothesis to the data scientist. And so we need at some point to actually, deconstruct the question and actually find some patterns that math guys can understand. And what is the nature of the data? What is the standardization of those data? It could be different types. You mentioned omics, you mentioned imaging, and all of those. And so those data actually have different fixtures, different parameters. Is the same for bioprocess as well. And so when we do this action, actually some technologies are needed to actually link the different data. And in order for the IT actually to build models on it, but also to extrapolate what are, let's say, the molecular space where the models are applicable. And that is really, really key in all those processes. The thing is, I guess that the field needs definitely to de-black box, actually, the ML models. So we know that there are different, uncertainties, but also we need to understand what the data means, where the models are applicable, and where actually we can help the different partners to move forward with. I will finish on that.
Good, thank you. Fabian, question for you. Can you talk about how you see digitalization supporting market growth opportunities for cell and gene therapies, such as new modalities or new geographies?
One of the key areas that I think is overlooked when you think about what digitization of manufacturing processes really enables is the expansion to other markets. Think about what it takes today, you know? You're a pharma company, you've got your drug approved by the FDA in the US, and as a next step, you need to go to Europe, you need to go to Asia, and address patient populations in Europe and Asia. What that takes today is you go over there, you find facilities the size of football fields, then you attempt to hire thousands of employees, then you got to train all of those employees. Training somebody on GMP methods takes about 6 months. Average retention time, 1.5 years. So you spend a third of your time training them, and 2/3 of the time actually do valuable work. Not to mention that the fact that we do have a dearth of cell therapy talent in our industry today, and even finding and hiring thousands of employees to do manufacturing in another part of the world is extremely difficult. So it's a multi-year ordeal with a lot of cost and a lot of risk attached to it. So here's how this changes. We're building out IDMO smart factories in Europe and Asia. We can onboard a process, automate and transfer a process onto the cell shuttle in about 6 months. And then every tech transfer thereafter from one cell shuttle to any other cell shuttle anywhere else in the world is instantaneous. Why? Because the entire manufacturing process is automated. So when we talk about tech transfer, we're taking tech transfer into the digital age, where we effectively copy over the database entry that defines the entire manufacturing process, and the cell shuttle in Europe or Asia will do the exact same thing that the cell shuttle in the US did. And by the time that we've got this network of facilities built out, when a new company comes to us, it's actually that fast, you know, because we already have the facilities in place. That's the vision in a few years, but the main point remains, you know, cell shuttle technology, because of the high degree of standardization, the high degree of automation, dramatically accelerates expansion to other markets and de-risks expansion to other markets. And the digital component in that is, of course, the fact that your process is now defined in a digital file, as opposed to hundreds of SOPs and operating instructions for you know, a small army of humans to execute your process at scale in another part of the world.
Great, thank you. So I'm going to ask a question for all the panelists here, if you guys could start, and maybe we'll start down on the end, Julien, with you. How is your company making prioritization decisions of investments for digital initiatives versus operational or scientific initiatives?
Actually, we listen our team because they know their work first. And the thing is, at the very beginning, when we decide to build WhiteLab Genomics, we decide to be agnostic in the term of disease, but we were asking ourselves, how can we be specialized in AI for gene and cell therapy? And so we know that in ComBio in general, there are multiple expertise, but what we want to actually push forward is how to integrate the knowledge of other fields, such as, you know, antibodies development, design, and everything. And actually, we thought that structural biology linked to ComBio, linked to genetics, is actually the key of success to actually partner with, you know, like people that develop different products. And so, to answer your question, let's say more precisely, the way so actually it's fine by finding good strategy at the very beginning, by asking to our customers first, what is the pain point, where we can help. And so that's actually where we find ourselves taking decision for technologies first.
Great. Tim, same question to you. How do you make that prioritization decision between digital initiatives or operational or scientific needs?
I mean, I think the vast majority of the computational work we're doing at Senti is in advance of, is meant to drive the scientific goals of the company. So I think for us, it's a little bit of a Darwinian process. Like if the computational stuff can't outperform what you can do through just like a random screen, we're not going to do it probably. Because for us, the goal is to make the best product possible, and how we get there ultimately is one more of sort of cost and timing rather than being purist about the approach we take. That being said, we have found, like I said, that many of these high throughput computational strategies have been really instrumental for us to achieve that next sort of step change in function. So I think for us, anytime we're starting a new project, it's really looking back at like, okay, what's our past experiences on using X, Y, and Z methods, ranging from manual to screening to computational-based approaches, or maybe some flavor of them together, and that is ultimately what drives our decision. It's very practical in that sense.
Kevin, same question to you.
What's interesting is sort of Ori from day one was planned to be a digitally native connected set of devices in this space. So in some ways, what we're having to do is build prioritization discussions on how to have those manufacturing platforms run offline. So if anything, what we sort of are constantly pulled to is, can a box run without an internet connection in a facility with no connectivity? So if anything, we have to keep making investments sort of in the past, because from day one, the platform was native sensor-based IoT, really operating like more of a smart device than sort of what has been standard. And we've been really having to make investments in making sure it will work in offline modes. With that, I think, going forward, what we continue to look at is how can we not only make the biology and the streaming data that comes out for it as useful and smart as possible, but actually have a smarter set of manufacturing devices out there that are always up, that we actually potentially know are going to have issues earlier, and how do we really sort of move the servicing support of those platforms from reactive to proactive in the platform.
We call that shift left.
That's right. Yep.
Great. Fabian, same question to you. How are you making those prioritization decisions at Cellares?
It's a great question. How do we prioritize investment into digitalization versus operational investments? And for us, those things they're integrated hand in hand. We obviously made a massive infrastructure investment. We've raised $355 million in less than 4 years, and it took a lot of capital to actually build the Cell Shuttle platform. And the Cell Shuttle platform was a big investment in automation, digital technologies, and that's really the source of all our operational efficiencies now, where Cell Shuttle reduces the cost of manufacturing by 50%, actually for the first time enables commercial scale manufacturing at the tens and hundreds of thousands of patient doses per year per drug. So the impact of Cellares is that over the course of the next decade, together with our partners, millions of patients will live that would have otherwise died. And that's enabled by the operational efficiencies of the investment in the infrastructure, the platform, the automation first, where in terms of operational efficiencies, we now get much lower cost of manufacturing, the ability to actually scale and produce 10 times more cell therapy doses with the same amount of people, same amount of floor space, and at the same time, reduce process failure rates by about 75%, reduce costs. So much more scalable, reliable, and cost-efficient manufacturing that's enabled in operation through a massive investment into digital technologies and automation that we've built in-house.
Great, thank you. So we have some time. We wanted to make sure we reserve 10, 15 minutes for questions from the audience. Anyone have any questions you'd like to pose to the panel? Yeah, up here front. There's a box, I think, that we'll bring up so that the online people can hear. I think they're going to bring it up. It's coming.
They're actually throwable.
Like a phone box. Yeah.
So,
football.
Up here at front.
Is this part of yours?
It's blue and it's...
Okay. So I applaud and I support what you guys are doing at Ori and at Cellares. I think it's great that we're moving forward with the technologies that you're bringing forward. However, I do have a couple of questions and want to get your opinion on them. What I have seen in this industry over the last few years is not so much a technological hurdle that we're trying to overcome, but more of a cultural and a organizational limitation that we put on ourselves in terms of development of the processes and the adoption of the technologies. And I heard what you said about instant tech transfer, which I guess in theory makes complete sense to me. However, my experiences with different sites, even of the same CDMO or manufacturing company, they like building their own processes at their local sites and not using a standardized approach. So, no disrespect, but it just seems like to me that there's more than just the technology that needs to make this work. We have to think about the processes and that we actually execute to develop, as well as the cultural and organizational perspective. So maybe you can
Yeah, I love
comment on that.
Yeah, and I love that question. So I'll just echo for the group. So how do you manage the cultural transformation that has to come along with the digital transformation?
So
Go ahead, yeah.
I'll go first. I love this question. This is one of the key things that we identified as well, and that was one of the key driving factors that ultimately led to the decision to become a contract manufacturer operating at global scale. It's actually much easier for us to do that internally because we control our own culture. We set the culture and we make it very easy, low risk, fast for our customers to then benefit from the technology. So rather than convincing pharma companies and biotechs to make a multi-million-dollar upfront investment into a new technology that they have to get trained on, have installed in their clean rooms, possibly build clean rooms to even accommodate, we're doing away with clean rooms, but to build manufacturing facilities to even accommodate a large system like the Cell Shuttle, rather than going the path of making that sale, we're saying, look, we've got the culture. We're technology adopters. We're going to be the first adopters of our own technology, operate and deploy it in IDMO smart factories around the world, and then just offer cell time manufacturing as a service where you pay only for what you use. You pay by the dose. You can test it out in very low cost, low risk partnership programs such as the Technology Adoption Partnership Program that we just signed 2 deals with Bristol Myers Squibb for, and several other biotechs. Lyell is a customer. They just joined the Technology Adoption Partnership Program. So because of the cultural hurdles that you've identified, this is a much easier path for us where we say, you don't have to take the risk. You don't have to adopt a new technology yourself. You don't have to get trained on it. You can just try it out. Let us do the work, and we'll do it for you. And it's much lower risk, much lower cost, much faster.
So as part of your core company strategy, you've made that. Other panelists where that outsourcing isn't what you're going to necessarily do, technology adoption or running your own labs. What's your experience?
It's a great question, and it's, I will constantly pick on life sciences, pharma in general, of running 10 plus years behind modern manufacturing of anywhere. Our head of digital always uses the phrase of dog fooding these things, which is how do you get in smaller successes to build confidence and then really start to think of these things as critical going forward. And I think it is a challenge for this industry of how do we get platforms in front of people that are user-friendly enough and sort of low enough cost that they can start to walk before they run in this space. And what we've seen is giving them back real-time data on what's happening in both the biology and in their manufacturing platform. They have light bulb moments when they start to see things go wrong. And those things have been actually sort of surprisingly simple. CO2 running out from a facility in the middle of a process, and us being able to real-time alert them as to the fact that this is going on before any of their other internal environmental monitoring and other systems have. We actually had one this summer where the HVAC in the facility died on a really warm day, and we were watching in culture temperatures rise to get somebody in there. So I think it's the opportunity to introduce actually some of these pieces into earlier stage PD and grow those people up into the commercial manufacturers and potentially even all the way back into academia, because typically what we've seen is the academics get hired by the pharma companies, they bring whatever they had with them there, and then those people become the commercial customers eventually. So we need to start early in the chain and get accessibility to them in a platform that's lower cost and comfortable to them to really build the skill set from the ground up.
Tim?
I would say on the research side, perhaps we have less of that issue. I've found actually most of our scientists really crave this. They realize the power of it, and they get frustrated, dealing with like 10 things that don't work. They rather let's just try a million things at once, and they realize they can't do that themselves. So I think on the earlier side of research, people are, more and more are appreciating the power of computational approaches, machine learning, as part of their workflow. And I don't think overall there's that much resistance to using it right now. Just because a machine invented it doesn't mean that I'm useless, basically. That's not the philosophy that we have.
Fabian, anything to add?
No, I totally agree with what Kevin and Fabian and Tim just said. Absolutely, to back to the fundamentals and teach the spirit of the industry is really the key, and also really to partner actually to change also the mentality. I mean, doing scales, testing more parameters, and integrate all the data that are coming from the different tests is really the key challenge to actually change the mentality in some way. Yeah, absolutely.
So as a technology provider, I will tell you culture change is one of the biggest things we face. And what we've learned is short-term little wins begins to build. So rather than trying to build an entire end-to-end workflow, build one protocol that gives some value back to that user. And I think you start finding they move you in the direction that you want to go. It may not be the one you originally planned, but in the end, we are there to service the person on the bench running the laboratory equipment, running the processes. And so I think, my advice is to the group is you start small. You may have a grand scheme, but you have to start small in order to build success behind the digital transformation.
Adding on to what Trent just said about building on small wins, that's the same approach that we're taking. Technology Adoption Partnership Program is effectively preclinical proof of concept. And then from there, if you like it and it worked, and we demonstrated we can run your process on the cell shuttle meeting all the critical quality attributes, then from there you move on to the clinic, then you move on to commercial scale manufacturing.
Question? Oh, yes, we have one up here.
So question for Fabian and Kevin. So I love the fact that your hardware and software are fully integrated, et cetera, but the thing is you're still, it's very much dependent on what the critical quality attributes are, and that's still a moving target. Potency, the definition, is still a moving target. So you're pretty much, well, I guess kind of locked into your hardware. I'm assuming there's some modularity there. So how do you keep your offering flexible to accommodate whatever's changing in the clinic?
So how do you manage this kind of perhaps more rigid digital infrastructure to allow the flexibility that needs to happen in order to hit your CQAs, which you may not know even at the beginning and may change?
They're not necessarily, nothing's set in stone, right? This is not your your antibody paradigm.
I love this question. Thank you for asking this. There's a misconception that because you have an integrated end-to-end platform, the system would somehow be rigid or inflexible. And that is one of the key innovations that we've achieved at Cellares, where we built an incredibly modular platform that fully automates the process, but also gives you a tremendous amount of flexibility. So we're reagent agnostic. You can load any reagent into the platform. Every key cell manufacturing process parameter is exposed. Not even, well, who are the two dominant providers of electroporators in the industry? Both companies lock down their electroporation parameters. They consider that their secret sauce. That's rigid, By comparison, our electroporator is actually outperforming the electroporators on the market in terms of electroporation efficiency, and it exposes all of the electroporation parameters: voltage, pulse length, single phasic versus biphasic, which buffer do you want to use. It's all open. It's all up for configuration. That's the level of flexibility that we're talking about, and that holds true for every single unit operation. So flexibility is built into the platform. Then on top of that, because we're vertically integrated and we've built the entire tech stack internally, we can go through an entire design, build, test cycle in about 1.5 years. Every 1.5 years, like that, like clockwork, we can come out with a new product, with a new optimized cell shuttle. So as the industry continues to evolve, different technologies become relevant, it's very easy for us to integrate that. And we've actually put the hooks for that into the platform already, where the single-use consumable cartridge that we have actually has a little bit of empty space reserved for the integration of new technologies, specifically the single-use consumable component of those technologies. And then the same is true on the Cell Shuttle platform itself, where there's 20 universal instrument bays where we can build the corresponding instrument to then interface with that particular technology that's now located on the cartridge. But it's integrated in a closed system because it's part of the internal fluidics in the cartridge, and it's fully automated because the entire process on the Cell Shuttle is automated via 2 robots that handle all of the material transfers internally.
Kevin, your thought?
Yeah, flexibility as we go through this, I think, is a critical one. And one of the things that we've been really working on is how do we actually help people fail faster as they go through these processes to start to understand what those critical quality attributes are and some of their performance metrics as well. I think there's 2 parts to it at Ori that we're looking at. Is one, a library of those digital protocols based on the modality, the tech that you're using to try to actually get you to a better starting point earlier. Just from the runs that we've been doing in-house, we're starting to learn, more and more for certain sets of combinations, what the best optimized protocol is going to look like, and really what are those tests that you want to be doing back end to see success or failure. The next one is flexibility in a platform so that as you learn those, you can really adopt and adjust process going forward. That's both equipment side flexibility as well as really moving from a paper-based protocol that people would say is more flexible, but ultimately, if you think about like the L7s of the world, you can go drop in whole new components of those things and run them in a controlled manner. So it's really adopting flexible and then figuring out those as early as possible.
Yeah, I think one of the kind of challenges that we've had on the digital transformation from my perspective is, many of our legacy software products have been built workflow first. So as you modify workflow, then you have to rebuild the underlying data structure because the workflow drives the data structure. And I think in order to really be flexible, we found if you start with the data first, then it enables you to have a flexible workflow. It requires more work up front. It requires you to understand the data model of your organization or of your operation. But those don't change that much. I'm growing cells, I'm in a factory, I've got a cell shuttle, or I've got a biotech, Ori biotech unit, or I've got an NGS sequencer. Like those primary data model components are fairly consistent across processes. What's really changes is that sequencing and the flavor of the workflows. And so, I think what we found is you can be flexible and yet compliant and in the same platform as long as you think about it from the data model first and the process then follows. And giving that process flexibility is key to be able to accommodate these changes. Any other questions? I have one more question. Oh yeah, you have a second question. Go. You want to grab the? No, sure, go ahead. There you go, thank you. It's a little early.
Okay, question and a challenge, okay? Each of you have represented your companies very well, presenting what you're doing. I think that's great. It needs to be moved forward from an individual company basis. But having done this stuff for many, many years, I'll call it that, in the pharma and biotech industry, I think we lost an opportunity in the pharma and biotech to deal with an ecosystem of collaboration across multiple players. In the cell and gene therapy, especially with autologous, we obviously need to deal with data management and integration across multiple players, not just the pharma, not just the manufacturing, but the supply chain logistics, as well as the apheresis centers, patient administration, et cetera. I guess I would ask or challenge you with how you would deal with a data management ecosystem approach as opposed to individual companies, because I've seen in my humble few years the need to share data between the hospitals, between the manufacturing, between the logistics for C of I, C of C, chain of condition, scheduling, you name it. And I guess I don't see one company or one organization fixing all this. I think it's a collaboration across multiple companies. And I say that respectfully to you because I think what you're doing is excellent, but I think you have to deal with it in a bigger way across the ecosystem.
That's fair. So, and I think part of it, and we talked about this in our prep for today's panel, is the cloud, right? And movement to the cloud and adoption of the cloud, because that is the first barrier to being able to share data, is getting it out of your firewalls, right? And so I guess one question I would have for the group is, how are you thinking about cloud, and what do you see as what needs to happen in order to enable better data collaboration, perhaps with that approach? Yeah, whoever wants to go first.
No, I'll go first and say I wholeheartedly agree with you. There's too much software that's required to build it all yourself, and also no purpose in reinventing the wheel. If there's a really good solution for something on the market, just go with that, like we've done with the integration of L7, for example. So when we're making buy versus build decisions for digital software platforms, we really look at, is it absolutely required? And when it comes, I think the answer for us, in our case, the answer was anything that's super closely related to the automation and the hardware. There we had to build custom software that tightly integrates with the hardware. Anything that's a little bit further out, further removed from the actual manufacturing process, the actual QC process, we integrate with best-in-class systems like L7. Deloitte's a partner. They've been helping us integrate a lot of different software systems into the stack.
Yeah, and I think it's two parts. One is being outrageously agnostic to those people in your world, and really having as many of those pre-baked partnerships done as possible, but better allowing for these pieces to slot in to an existing ecosystem that may be multiple parts. And one of the things that we do talk a lot about is starting to digitize what people would consider the last mile. So whether that's automation platforms, making these things play at the larger scale with the Delta V's and Ignitions of the world, at the smaller ones down to the early ELN providers, and then for the people who are going the routes of the L7s of the world, is just making sure these platforms plug and play with as many of them as possible, and really don't constrain people making these decisions to any other part of the stack, right? I mean, it is a plug for Ori, but what I would say is the one thing that you will not see us doing is saying, oh, to get all these other capabilities, you now need to buy a stack of other software that's proprietary to our platform. We're not really in the software space. So we're happy to have those continue to exist and live in this sort of broader set of ecosystem, and let people bring what they have and then work with it.
Tim, Fabian, we got about a couple seconds
38
here.
seconds.
To, 15, 20 seconds just on the research side.
I would say on the research side,
that's
data silos are a frustrating thing. I think the reason why technologies like AlphaFold were able to achieve what they were able to achieve is there's an open competition with open data sets that everyone had access to, right? And that's why people could compete on demonstrating technology. I mean, in pharma and biotech, people don't share stuff at this point. And so I don't know how to encourage that, other than maybe you can come up with some consortia where if you contribute data, you get something out of it. Yeah. An open license to the outcomes of that data or something like that. I know certainly in academia, NIH and others are trying to force more and more open data sharing, but enforcement of that is also difficult. But that's one of the things we spend so much money as a field investing into discovery, and then something fails, it just sits there and no one ever knows what happened. So that's a frustrating thing for people who are trying to develop new drugs, for sure.
Fabian? Yep. Okay, good.
So I think to answer your question, we need to change the question itself. Because you talk about sharing data, but it could be how to share models to develop something, how to also investigate the data itself. You know, in every byte that's been transferred to a partner, actually maybe just 30% of it is actually useful. So that is important also. All along the analysis of the data, when you do the structuration, when you do the harmonization, when you do the QC, everything. So the thing, what you observe is actually to your partner, not all the data is necessary. And so you need to transform also those network in order to actually understand what you transfer, what is useful, how to use it. And I mean, the infrastructure, I mean, AWS, Google, et cetera, have all the knowledge to do the network. But then if you want to learn from the data, if you want to develop new innovation, if you want to develop new technology, there are in machine learning, in other, like either in the database technology, such as knowledge graph database, or in federative learning, there is technologies that actually can move, make the field move forward with more innovation about it. And maybe, that's one of the points that you mentioned, Kevin, is that the field is do not share data, but you don't need to share data. You need to share the will to actually work together and without copying the data. And that is possible through new technologies. And if you want to talk about that, in WhiteLab, we definitely, like moving in this direction. So don't hesitate to come to us.
Super. Well, first of all, thank you to all of our panelists. It's been great, and I think a great kickoff to this year's MESA meeting. And to all of you for coming early, bright and early. I hope you have a great rest of your conference.